Hello everyone, and welcome to today's webinar, How the New FDA Guidance on ISO 10993 Could Possibly Affect You, brought to you by Nelson Laboratories in partnership with MD&DI. I'm Shana Leonard, Executive Editor of the UBM Canon MedTech Group, and I'll be your moderator for today's event. Our speaker today will be addressing the draft guidance released by FDA in April on biocompatibility testing for medical devices. Before I introduce our speaker, however, I do just have a few brief general announcements, so please bear with me. This webinar is designed to be interactive, so please feel free to submit a question at any time during the presentation using the button on your screen. After our speakers finish his presentation, we'll conclude with a live Q&A, and we'll try to get to as many of your questions as time permits. And now that we do have those housekeeping items taken care of, we're ready for the main event. So, joining me today to discuss the new draft guidance on biocompatibility testing is Thor Rollins, the in vivo biocompatibility section leader at Nelson Laboratories. Thor is a certified microbiologist who has been with Nelson Labs since 2002 and is an active member of a number of groups and committees focused on biocompatibility. Thor, thanks for being here, and the floor is yours.
Thank you so much for that, Shana. And I'm really excited to be here today with the webinar. Usually with webinars, I get to talk about certain tests or certain standards and how they might affect devices going forward. But this is the first time that we've really got to be able to talk about some guidance given by the FDA that could affect medical devices. In fact, this draft document that's released is meant to replace the G95 document that was released in 1995. So we haven't really seen any kind of guidance at this magnitude for over 18 years. So that's why this is quite exciting. For those of you that have not seen the guidance document yet or have not had a chance to read through it, the first slide here is just kind of a link for you to be able to go and look at the guidance document, kind of read through it, to be able to get comfortable with it. So this is the first page of the guidance document, and as was mentioned, it was released in April. The timing was actually kind of funny. The release came right during the ISO 10993 committee meetings in Pavia, Italy. So we were at Italy at the time, and this was released, and so we spent a lot of the time in Pavia actually going through this, and in fact, the ISO committee kind of organized a lot of their comments together and submitted them. As you can see, we had 30 days to submit comments for the draft document, and so they were submitted to the FDA to try to get some clarifications. But all in all, I want to stress that I think this document is a very good document. I think there's some really good information that we've seen come from this document, and I kind of want to share some of those thoughts with you. In fact, here at Nelson Labs, what we did is we combed through that document, and we've broken it down a bunch of different ideas into 3 categories. Now, these categories are not set up in the document, these headlines, but it's kind of our translation or our thoughts on the document. And we break them up into summary of ideas, justification ideas, and possible impact items. Summary ideas are these ideas that we've actually seen from the FDA for a couple years now, maybe even longer in some aspects, but we've never had them actually written down other than response letters from the FDA. But now we actually have them written down on a guidance document that we can reference, we can point to, and so that kind of helps with either when you're looking at setting up a test design or when you're looking at the impact of a failure, for example. There are some ideas in that category that have a big impact there, but these are ideas that most of us who have been doing biocompatibility have seen recently. The next category is justification ideas, and this is, for me, one of the most exciting areas. Because in the past, sometimes it feels like when you submit, it's been kind of a checkbox mentality where it doesn't matter the type of the device or the contact or duration or the materials you're using. The G95 was used as kind of a checkbox. But now there's some great ideas in there laid out about what to look for as far as testing goes, how to change materials or how to look at the testing more in a scientific point of view and just not go through the checkbox. It does put some more strength on scientific analysis on these tests, and so it does require a little bit more knowledge and background on the tests themselves. But there are some really good ideas in there on how to justify biocompatibility, specifically animal testing, and look at ways that we can limit the amount of those tests for certain devices. Then the last category, which probably is the most impactful to manufacturers, are issues that we have not seen before or questions that we might have had either as an ISO group or as Nelson Labs independently that we think might have an impact going forward that we have not seen the FDA stress up to this point. So I'm going to go through each one of these categories. There are some things that I am going to explain a little bit to give you a little bit background knowledge on how to do that type of justification, for example, or testing. But for the most part, we are just going to go through certain topics on the guidance document. So the first thing we're going to touch is that summary of ideas. And once again, to remind you, these are the thoughts or the ideas that we've seen from the FDA previously, but we've never had a reference point for these thoughts. So the first one is the document incorporates new ideas regarding toxic chemicals, and specifically they mention colorants. And obviously this was not addressed in the G95 document. This is more hanging around the ISO 10993-17 and 18 with the chemical characterization, where we're using chemicals and what comes off the device to be able to address safety concerns. And this might help limit biocompatibility testing depending on how it's used and where it's used. But the FDA has always had a kind of a specific, or recently has had a specific concern about colors. Color additives have known to be potential carcinogenic or genotoxic, and so we've had questions come back from devices that have colors in it on the FDA wanting to see how much colors leach off the device and how much of that colorant interacts with the body and what's the safety impact from a toxicological assessment. Now, there's two ways to kind of do this. The first way we've seen requests from the FDA is actually perform a leachable study where we mimic the clinical use of the device and actually look for specific colorants that are coming off and then address those amounts. The second way is to kind of limit the leachable study, and we just say this is the total amount of that colorant in the device. If all that colorant was to release at once, this is the impact from a toxicological assessment. So we've seen both options be successful, but this is, once again, the first time we've seen it kind of addressed. And here in the next slide, we'll actually kind of talk about ways that we can look at the chemistry to address it and kind of how to do that chemistry. The next one is sponsors are advised to discuss test plan with the FDA before initiating testing. And this is something that we've stressed for a very long time here at Nelson Labs. These biocompatibility tests are so long and so expensive that it's nice to go into the testing with kind of a confidence that as long as the testing is performed correctly and the results are good, that you're doing the correct test plan. So this is a way to get feedback from the FDA on specific tests, and you have limited opportunity for surprises when you actually submit those tests at the back end. The last one here on this slide is the FDA does not approve individual materials, but only final devices. And this goes to the concept of processing, and we'll actually see this later on. But a lot of times manufacturers will try to take the raw material, and they'll have biocompatibility information on the raw materials, and they'll try to make assumptions on those raw materials on their final devices. I'm not saying that can't be successful, but you really have to evaluate what processing you're doing to those materials in your final device. So is there any kind of molding that could have mold release agents, or sterilization can have an impact? Specifically, gamma sterilization tends to have a big impact potentially on biocompatibility. So just because your raw materials have a good information doesn't necessarily mean that you can eliminate all your biocompatibility. But there is, when we get to the justification area, there are kind of aspects that you can incorporate some of those materials or some of that information to reduce the animal testing. So we'll talk about that in a little bit. So first, I kind of just wanted to hit that material characterization. I know that we mentioned it as a way for colorants. We use it for everything that could be extracted off a device. And so here I have a real-life example for chloroform. But if you're looking at a colorant, then all you'd have to do is kind of replace chloroform with that specific colorant. So in this example, the device extracted off 0.00023 milligrams of chloroform off the device. So how we look to evaluate safety is we do something called a human daily exposure, where we take that amount and we divide it by 70 kilograms of body weight. That's for a general population, that's 70 kilograms. If your device is patient specific, so if you're looking at neonatal or infants or even women, then you want to use a corresponding weight for that population. But basically what you're trying to determine is the dose of that off that device or that chemical per body weight. So in this case, we got 3.3 times 10 to the negative 6 milligrams per kilogram of compound, or body weight, excuse me. So now that we have that daily exposure, we can do a quick kind of calculation where we get some toxicological data. We're looking for NOELs or LOELs. They're just basically levels where we see no observed adverse effects of that compound in clinical data. And then we might have to times that human dose effect by some modifying factor depending on the quality and type of data we get. So to give you kind of a real-life example, this is ToxNet. This is a good resource. There are other ones. EPA is also a great resource. They're anywhere that has toxicological information on compounds. But we go into the website and we put chloroform up on the line where we're looking for that specific compound. And if you have a colorant, then you might have a CAS number, a CAS number for that colorant, and you can actually put that CAS number into that search line, and that will also pull up the data. But basically what you're looking for is specific data on that compound. And in this aspect, in this example, we went to ToxLine as our resource for that data, and we had a ton of data that we got from chloroform that we could go through. And so what you do then is you go through specific testing from this database looking for the compound. And in here we found some tests that had ranges of NOELs, that's the no observed adverse effect levels, between 15 and 125. So what we're trying to do is determine which of these levels is the best representative of our device. So which one has the most representative contact to the patient, the same route of administration and dwell time. And that's the NOEL we want to use. In this case, in this real-life case, the NOEL of 15 was the most realistic. So we could plug 15 into our mathematical equation where it's 15 milligrams per kilogram. That's the NOEL. The 3.3 times 10 to the negative 6 is our HDE, the exposure. And then we did a 100 safety factor, and that was just based on interspecies and intraspecies differentiation, and then also maybe quality of data. And what that gives us is a margin of safety of 45,000. So what that realistically means is that in this device, you could have 45,000 times more chloroform come off the device compared to that NOEL and still be considered safe. So this is the kind of mathematical and research you can do to help justify chemicals that are coming off your device, specifically with colorants as a way to evaluate colorants. So this is a good way for us to kind of look at a risk assessment of these devices, specifically with the draft document and what we're seeing from the FDA. So going on to more summary of ideas, one of the other things that they talked about is that if you have a certain target organ for your device, and they specifically mentioned neurotoxicity, so if your device is meant to go into the brain, then we have to do testing that's specifically targeting that organ. Now, for most medical devices, this is not a big concern. Most of them don't have a target organ, but we have had instances recently where we're using a neuro device that we'll have to do either neuroimplantation with toxicity involved, but we do have to be aware of any target organs that the device or your device may be interacting with. The next concept is master files are not sufficient for submission because they do not incorporate processing. Now, I told you processing was going to come up again, but here it is. So once again, we hear from clients all the time that all their materials have master files and that they don't have to do biocompatibility. Now, master files can help in a risk assessment. In fact, they help a lot in a risk assessment. But you cannot just submit master files and think you have a checkbox with your biocompatibility because those master files may not have been performed on material that had gone through the same processing as your final device. That's including sterilization. So if your final device is sterile, these master files may have not been tested on a sterile device or a sterile material. So you have to evaluate the impact of your processing to these materials before you can make any kind of risk assessment to get rid of biocompatibility. The next topic is in vivo and in vitro testing should be conducted GLP. Now, I'm not going to spend a lot of time on that because if you're really spending this much time and money on biocompatibility, the small increase in GLP costs should be worth it. For those of you who are not sure what GLP means, it stands for Good Laboratory Practices. It's kind of a quality built into that test. The testing is performed exactly the same, but it's audited by an independent quality group. And so the FDA prefers to see the GLP tests performed for biocompatibility. The next set of ideas that are from the FDA that we've seen in the past is that for permanent devices, you should also perform or need to justify chronic toxicity and carcinogenicity testing. In the past, when the G95 came out, carcinogenicity testing really was not even evaluated for most devices. Genotoxicity was used to try to justify out of, but now with the extractable leachable testing that I just talked about, we can actually evaluate both chronic toxicity and carcinogenicity with those compounds coming off a device. So we can use the toxicological information we get from those compounds to look at those. So with an addition of the genotoxicity testing and some of the subacute or subchronic toxicity testing, we can actually look at justifying out of the chronic toxicity and carcinogenicity. And once again, once we get to justification ideas, we'll kind of talk about that in a minute. The next one is guidance on representative coupon devices. And I love this one. Many devices are very, very small or very expensive to manufacture. And for biocompatibility, we only care about materials and processing. We don't need to test a functional device. So we have guidance now in this document on how to develop coupons or representative testing devices. So these testing devices are devices that represent the same material in the same proportions of your final device and the same processing are represented. So basically we can take whatever material you have in your device and make it into a coupon, which is maybe a square or some kind of thin sheet, something that might represent your final device, but we don't need to test a functional device. This is also beneficial for devices that have internal components that are not patient contacting. So electronic components, other wires, things like that that may be in the device, but we don't want to necessarily test them. We can actually test a coupon that does not incorporate those materials. The chart of test consideration is not intended to be a checklist, and this just makes me smile because once again I mentioned before that sometimes in the past the G95 was used as a checklist. So when the reviewer or even the company was evaluating what biocompatibility needs to be done for the device, they just kind of went down and did the testing on that list. That's not really the intent of these documents. The intent is to kind of show you what categories are of a concern for a contact or duration of a device. And then you have to scientifically look at those and justify which ones need to be done for your device. And that's kind of what this guidance document now incorporates also, that this chart they give you is not a checkbox, but is a way for you to evaluate what testing is needed for certain devices. So the next summary of ideas are possible device failure options. So a good example here is a balloon on either a device that is a cement or some other balloon functional device where the balloon inside the balloon, the components inside the balloon are not intended to have patient contact. But if there's a failure of that balloon, if something happens to that device that it fails and exposes part of that device to the patient, then we also have to evaluate those portions of the device for biocompatibility. There are different ways to do this. ways to maybe limit the amount of testing. We can look at the differences between inside and outside. We can look at the surface area compared to the whole device. So there are some good scientific ways to look at this, but you have to evaluate possible failure options of your device. The next one is with the sample preparation. So for biocompatibility, we've always looked at two ratios of how to get the device into a solution, how to extract it. And those have been based off either the surface area device, so the larger the surface area, the more volume we add to the device, or by weight. The heavier it is, the more volume we add. And it's been kind of ambiguous in the standard on which one to use. Now, recently the FDA has been very strong on using the surface area, and once again they mention here that surface area should be needed. The next one, and after this slide I'll kind of show you an example why that is, but just keep that in your mind for right now. The next concept is for prolonged contact device extractions should be performed at 50 degrees unless sample chemically changes around 37 degrees. So in this case, because it's prolonged contact, the extraction parameters in the standard is 37 degrees for 72 hours. If your device has longer than 72-hour contact, it cannot mimic that prolonged exposure to the body at body temperature. So the FDA has been asking us to elevate the temperature to get a prolonged exposure. My suggestion is that unless your device changes chemically at 37, just do 50 regardless of duration. Because it's worst case, most devices will handle 50 degrees Celsius well, and you don't have to worry about trying to answer that 37-degree question. Okay, so I talked about the difference between surface area and weight, and here I have an example to show you. So here is a partial knee implant. They're made from all sorts of different metals, titanium, stainless steel. But when we look at this device, it weighs 93.9 grams. So if we use the weight to volume ratio in the standard, that means we would extract this device in almost 500 mils of media. So we would set this device into half a liter of media and extract it. And that's based on the weight ratio. If we look at its surface area is about 115 centimeters squared. Now it's thicker than 0.5 millimeters, so we use the ratio of 3 centimeters squared per mil. This gives us the 38.6 mils of extraction fluid. So much less fluid being extracted or used to extract this device. So when we look at a comparison between the two ratios, weight gave us 468 mils, surface area gave us 38 mils. So with weight, we actually had 12 times more media used. That means those results would be 12 times diluted because all the chemical substances being extracted would be suspended in that solution. So we would be much less sensitive using weight compared to surface area. And that's why the FDA is preferring the surface area for extractions. If you use weight, you have to justify why you used it, and I can guarantee you that while it required less sample, it's not a good justification. Okay, so one last thing I want to kind of talk about is that we have an evaluation of when you fail a test. And this gets into our next slide a little bit. But I wanted to give you a flow chart that I use to evaluate a failure. Because with biocompatibility, you always have to worry about failing, specifically the cytotoxicity. So this flow chart kind of goes through a step-by-step procedure on how to evaluate a failure. And this goes into the justification ideas here in a minute. So I'm just going to cover this very quickly. But first we want to confirm the procedure. We will make sure that the test was done correctly and the protocols were followed. Then we want to confirm the result. So here you might want to break down into individual components or take a look at why, or make sure that substance tests are also failing. Next, once we know that there's something toxic on the device, then we want to try to eliminate the toxicity. But then there's another option where we can look at accepting the toxicity. And this is where I want to kind of jump into, which is a risk assessment. In this new draft document, we actually kind of give guidance on looking at accepting toxicity or accepting the device results of a test based on the risk assessment to the patient. So how is the patient's health impacted by this device? And will the lack of the device hurt the patient more than what maybe a cytotoxicity result would give? So going back to the summary of ideas, The first one here is no manipulation of the extract. So we talked about the extract on the device by weight or surface area. After we have that, the FDA does not want us either filtering or centrifuging those extracts that we may get rid of any particulates or chemicals that are in that. So make sure if you are testing at a lab that your lab does not manipulate those extracts in any way. This next one is actually quite interesting. It's samples for cytotoxicity should be extracted in cell culture media with 5% serum. Now with cytotoxicity, we extract in not a strict polar or strict non-polar, but we extract in something called MEM fluid. It's a cell culture media. That MEM fluid usually contains 5% calf serum. But that 5% calf serum has always been kind of a suggestion before through the industry, and now we actually have it put into a guidance document from the FDA. The reason why this is important is because sometimes some labs are extracting in saline, and then they use the saline to test. Now, saline is not supportive of cell health. That means you can't put a cell or saline onto cells and have the cell survive. So they have to mix the saline into MEM fluid on the back end. So what's happening there is your neat solution or your 100% pure extract is getting mixed into an MEM and is getting diluted compared to when we extract straight into the calf serum or the culture media with 5% calf serum. So when you're doing a cytotoxicity test, make sure your lab is performing the test with a 5% calf serum. It can have an impact on results, and the FDA prefers the 5%. When you're looking at the next category, when you're looking at the blood contacting devices, this is kind of this is an old feeling from the FDA. We are actually looking at rewriting ISO 10993-4 to try to incorporate some of this. And in fact, the author of this document, or one of the authors of this document, Jennifer Good, is on the -4 that is helping us write. But it's stated in there that for blood contacting devices, regardless of duration, they want you to do hemolysis, complement, and thrombogenicity testing. Now we're going to talk about a little bit in the justification section on how to maybe look at justifying out of those tests, but right now that's the statement. The next category is intramuscular implants are preferred over subcutaneous implants. This is just the implantation. The device is the preferred method per this guidance document is to actually implant those devices intramuscular. We do that in a rabbit, so we do the implant along the spine of the animal in its muscle tissue. There's another option where we can do it in a subcutaneous space, but, and I agree with the FDA here, it does have a lower sensitivity compared to the intramuscular for most devices. So in that case, the intramuscular implant is preferred. Okay, so now that we've kind of hit all the ideas that the FDA has already kind of expressed, but never really written down in one place, we're going to move on to some ideas that can help you justify different testing. The first one is weigh the benefit of the device to any potential risk associated. So once again, we talked about this before, a failure justification. When you're looking at biocompatibility, just because you fail cytotoxicity does not mean that your device should not be used. You really have to weigh the benefit of the device of that failure. When you're looking at chemotherapy drugs is the greatest example. Chemotherapy drugs are highly toxic, but when you look at the benefit compared to the risk, then they are able to be used. The next little point is reviewers are encouraged to apply scientific judgment when reviewing alternative data from animal tests. Once again, these are, if I could put little smiley faces by each comment, this would be one that I would do. I feel like there are a lot of great tests out there that you can be used, in vitro tests or chemical characterization tests that can be used to help justify some of these animal tests that have been around for a long time but may not have a great equivalence to the actual impact to the patient. So here the reviewers are encouraged to apply scientific judgment. Now, the other side of that is you as manufacturers are encouraged to apply scientific judgment on what tests you perform. That might be a little harder. This is where you might need to contact someone who is maybe a biocompatibility expert or someone who's had experience in this industry to really go through your test development, to be able to look on where you're able to maybe use that scientific judgment to get out of doing some of the testing based on your device. At this point, I can't give you a general guidance because each sample is so specific, each device is so specific on this that it really is a case-by-case basis. The next step is literature can be used to justify out a systemic toxicity test, but the correct data has to be used at the corresponding endpoint. Once again, same kind of concept. Here's scientific judgment being used to justify out of testing. But you have to have someone who can know the correct endpoints of those tox tests and make sure that the data you're deriving has the same endpoints to be able to make those justifications. The next one is biocompatibility testing and or adequate chemical characterization in conjunction with supplementary biocompatibility testing may be acceptable to minimize animal testing. Same general concept. Here, though, we have, once again, other biocompatibility, so some in vitro tests, and we talked about the chemical characterization, may be able to minimize animal testing. Another smiley face there. The next one is if the intended use, physical form, formulation, processing, component interaction, and storage conditions are the same as a predicate sample, that no/limited testing would be required. And this one also kind of makes me smile, not because I'm thinking that this is going to be great for actual submissions, but a lot of times I get my clients call in and say, I have a device that's the same material as my competitor device, and they have a 510K, so I'm not going to do any biocompatibility. That's not where that stops. If you look through these checkpoints, there's a lot of things you have to consider when you're looking at saying no biocompatibility is required based on predicate or other use. So these are some of the checkpoints you have to look through to kind of make that assessment. The next one is actually something that we talked about a little bit with the hemocompatibility test, but we talk about functionality tests can be designed to look for some of the endpoints of the biocompatibility testing to help reduce animal tests. And this we see most successful with the hemocompatibility. A lot of times these devices are tested using functionality tests in large animals where they're actually looking to see how the device performs. We can incorporate a lot of the hemocompatibility endpoints into those tests to help eliminate some of the animal tests in hemocompatibility. There are some other specifics also with implantation, chronic toxicity that we can also look at. What we need to do here, though, is you need to have a good conversation with a biocompatibility person before you start those functionality testing so that we can actually incorporate those endpoints into the functionality protocol to be able to help justify out of the testing at the back end. Okay, so now the final category we're going to hit is possible impacts to you. So once again, these are concepts or ideas that kind of stood out to us as changes that we have never seen before. And each one of these, we've either have responded as an ISO group together or we've responded to them as Nelson Labs independently. And I'll kind of comment on how we responded or what kind of discussion was on each one of these. So the first one is that devices that have multiple types of exposure should consider testing from both categories for your device. Now, this example off the top of my head for this one is that if you have, let's say, a drug delivery device or even an IV bag, for example, and you have some kind of port that's left into the patient that might have different devices going through, and that port is implanted for long term, but the devices have short current contact, then you have to evaluate both categories of your device. In the past, some device manufacturers have just incorporated components of a shorter duration in with all their long-term duration tests, saying it's worst case they're going to get the long-term tests. But what the potential impact of that is we base all this test off surface area. So the more surface area, the more volume we add. So if you're combining components of that device that do not have long-term contact in with the device that does, then you're increasing the surface area of all the material there. So let's say some of that device that does not have long-term contact is toxic non-toxic, but the one that has long-term contact is toxic. By increasing the surface area, you could be hiding those results. So this is why it's important to kind of separate the two contact out so that you're not hiding any toxic materials by increasing the surface area of safe materials. The next one is right along the same lines as that, is devices that include components with different lengths of contact. Then here they give you an example: implant and delivery system. Conduct extract tests on components separately. Same concept, but here they actually give us a specific example, a stent in a delivery device. You cannot include its delivery device in with all the stent testing. This next one is one that's very minute, I guess you can say, almost, I don't know if it was intentional or not, and that's the question that we're going to we gave to the FDA. So in the document, and this is straight from it, it says implants, comma, devices in contact with cardiovascular, comma, lymphatic, comma, or cerebrospinal fluid, comma, or labeled as non-pyogenic need to perform the LAL test. Now, in the past, we've always had guidance from, especially from USP, that says that implants that have those contact types have to do the LAL test. Now, if you're not familiar with the LAL test, it's a pyrogenicity test. It looks for a fever, but specifically looks for a pyrogen called endotoxin. It's a compound found in gram-negative bacteria cell wall. In this document, there's a comma after implants. So for me, in my interpretation, that implies that implants, all implants, and then devices with those contacts have to have the LAL test. I'm not sure if that's exactly what is intended in this draft. Our feedback in this area is mentioned that either we need to have some clarification that all implants, even orthopedics, have to have LAL tests performed on them, or are they just specifically saying devices that have those implants, once again, with those contact types? So that's something that's still up in the air. We have not gotten feedback or response from our questions yet, but that is something that we have our eyes on going forward. I can see interpretation both ways, but we want to make sure that we're interpreting that correctly. The next one is all rabbit pyrogen tests need to be extracted at 50 degrees C or higher, regardless of contact duration. Now, this one's kind of funny to me because we did have one earlier where we talked about prolonged contact need to be 50 degrees Celsius. This one just says all rabbit pyrogen tests need to be extracted at 50 degrees. I'm okay from a scientific point of view saying that, but the one thing, and this is my feedback to the FDA on the draft document, was what about those devices that have chemical change at 37? We mentioned that before in the kind of principles in the first part of the presentation. Here we don't have that kind of out, where if the device changes chemically. For me specifically, there are some compounds that at 50 degrees, they might actually start to degrade and become more viscous in solution. And we're, once again, not really allowed to manipulate as far as centrifuge or filter that extract. And we all know that, well, if injecting maybe a slightly viscous solution IV into a rabbit, we could have some material pyrogen that's not actually from the device, but just from a mechanical reaction from that extract. So we're asking for some clarification there that if the device changes physically or chemically at 50 degrees, can we still use a 37 degrees for the rabbit pyrogen test? The next one comes with genotoxicity, and this is based on some historical data that we've seen from the FDA preferring the animal test, the mouse micronucleus, along with the Ames, and then you get to choose either the mouse lymphoma or the chromosome aberration. Now, in this draft document, they actually come out and say the mouse lymphoma test is preferred since it detects the broadest set of genotoxic mechanisms. We actually have some contradicted data, some evidence that we submitted into the draft feedback that actually suggests that the chromosome AB actually can detect a broader sense of genotoxic mechanisms. In fact, Japan in the Berlin ISO meeting about 3 years ago presented some good data that the chromosome AB might be the most sensitive genotoxicity test. They were trying to push that the chromosome aberration be considered the most preferred genotoxicity test. Now, so we're asking for some guidance with that statement as far as the mouse lymphoma test is preferred over the chromosome AB. The next one has to do with the chart, and actually this is the last possible impact, but it's one that might have the greatest aspect of impact, and that is they changed the chart in the G95. The old chart, if it has diamonds, so it has little dots of the tests that are required, and then you have diamonds on some other tests. And the diamonds state that additional tests may be applicable. So basically it gives the FDA reviewer allowance to ask for those tests depending on your device. Now I really think that this is still going to be the same, but they changed the verbiage on this draft to say these additional tests should be addressed in the submission either by inclusion of the testing or a rationale for its omission. So obviously this puts a higher emphasis on these diamond tests. Now we did talk about earlier in the justification realm that that this should not be a checklist, that you need to evaluate each one of these. And I think this is kind of a piggyback on to that, where the diamonds should be assessed, that you should look at them and really address they should be addressed in the submission either by justification or testing. Once again, I really want to put an emphasis that this draft document, in my opinion, puts a more emphasis on you knowing the scientific rationale behind the testing, looking at the device, and really coming up with a meaningful test outline for your device. And I think this would probably have to have more interaction with the labs, with biocompatibility experts, if you do not feel comfortable having this discussion or this exercise by yourself. I would be happy to help anyone who has ideas on how to do this or questions. My contact information is in the presentation. Feel free to give me a call, shoot me an email, go to Nelson Labs website if you have any questions. We'd be happy to help. So at this time, I did put in the last slide, I did put the actual chart from the guidance document or the draft document where they mention that, so you can actually have a reference if you don't have the draft document in front of you. Okay, so at this time we're going to go ahead for questions. I also wanted to thank you for your time and your attendance. It's like I said, this is something that's very exciting for me, something that I'm glad that I got the chance to talk about. But I'd like to turn the time over back to Shanna for the question and answer portion.
Thanks, Thor. That was great and very insightful. And it seems like our audience is really engaged. We already have questions pouring in. And at this time, I would like to open up the floor to your questions. If there are any additional questions, please remember that you can still submit questions, and we'll try to get to as many of your questions as time allows. However, as I mentioned, we already have them pouring in, so that's great. We will start off with one that asks, what about short-term intraocular devices? Do they require the rabbit pyrogen testing?
That's a great question. So we have actually seen a higher emphasis on any kind of ocular contact, specifically with pyrogenicity. So with an ocular contact, you already have the endotoxin test, but we have seen where and at the top of my head, I cannot remember if it was the FDA or if it was an international regulatory body, but we have seen emphasis where they want to see a rabbit pyrogen. Now, in my opinion, the rabbit pyrogen test really isn't a great predictor of pyrogenicity. It takes a lot to cause a response. It's not a test that fails very often. But when you're a reviewer and you're worried about possible pyrogenicity from an ocular contact device, we have seen that kind of thought that they want to see additional pyrogenicity. So even though in this draft document, in my opinion, it doesn't have that. It has a diamond, if I remember correctly, in the chart. So you do have to address it, but it doesn't necessarily mean it has to be done.
Okay, thanks, Thor. Our next question is repeated exposure considered prolonged exposure, for example, 8 hours a day, every day?
Great question. So in the concept of ISO or biocompatibility, we look at the total additional time of a device for its contact duration. So when you're looking at maybe an IV bag in the hospital, let's say a patient only sees an IV bag for an hour at a time, but that duration of time, that patient could be in the hospital for a month. You have to add up that time together for your total contact duration. So just because you only have an 8-hour window doesn't mean you're only limited contact. If that 8 hours starts to add up over time, that's your total duration of contact to the body.
Okay, great. Moving on to the next question. Somebody writes in that they're an OEM and their process and materials have not changed, but their supplier has. Do they still need to repeat their biocompatibility testing?
This is a question I get asked probably 2 or 3 times a day in different kind of forms. And it really depends on the, how sure you are of your new manufacturer or your new supplier of material. What I usually recommend is there's some great chemistry tests to evaluate how similar those materials are. Very inexpensive and quick. So we can actually set up kind of a testing, quick and cheap testing outline to be able to help back up your acceptance that those materials are the same. I always prefer to have some kind of risk assessment or some kind of evaluation when you're changing manufacturers or you're changing suppliers, even if you swear or they swear to you that it's the same material, just to make sure you have something to back you up. The cytotoxicity test, for example, is a great screening test for this, along with some of those chemistry tests. So I would still do some, but I don't, depending on the type of change and the results from those tests, you might not have to do all your biocompatibility.
Okay, great, thanks. Our next question is, which hemo tests are considered as being a thrombogenic test? Do prothrombin time and unactivated partial thromboplastin time tests meet this requirement?
Yeah, that is $1 million question. So it really, those are our thrombin tests. And it really depends on the information you can give the FDA regarding thrombosis. The test that everyone wants to try to get away from performing is the in vivo or the dog thrombosis. That test is, in my opinion, not a very good test. We see when we have 2 dogs, we might see one dog have thrombus on the test article and on the control, and then the other one neither. So it's not the great predictive test, but it's the only animal test that we have that can really give any kind of emphasis on how the device is being used, other than just with circulating blood with a dynamic flow system at this point. There are some other in vitro tests out there that we're considering that might mimic that, but at this point, that's what we have. Now, if you're able to look at thrombosis with those endpoints in functionality tests or some other tests, then you can use some of those in vitro tests to help justify. That's where that whole use functionality test to help meet endpoints comes in. But right now, if you're looking at thrombosis, the dog thrombo, if you haven't done any other work, the dog thrombo really is the test that some reviewers are looking for to checkbox thrombogenicity.
Okay, great. Thanks, Thor. Again, we have questions pouring in, which is great. Our next question is, have you had conversations with FDA to provide support to your opinion that the new guidance indicates a shift from a checklist mentality at FDA? In reading the guidance, this person feels that there will be increased testing required as opposed to less.
Well, and there are aspects of this draft, like I mentioned, the chart, where there could be increased testing. That being said, all the conversations I've had with reviewers, specifically with some of the reviewers that are authored this document, is they do understand that they want to get to a checkbox mentality, or excuse me, away from a checkbox mentality. They do want to get to a scientific rationale, and they are trying to encourage their reviewers to go that route. Now, the hard thing about the process is that you are dependent on your individual reviewer and their support system when it comes to scientific rationale. This is why, in my opinion, it's even more essential that you come up with a good test plan with whoever you feel comfortable with, submit that to the reviewer beforehand to really get an idea of what their background is, get some communication with a biocompatibility expert at the agency to be able to understand what they feel is scientifically justified compared to what your feelings of scientifically justified are. So I do agree that there are aspects of this guidance document that could encourage more testing. I hope that there are other aspects that you can point to really try to eliminate specifically the animal tests, specifically in those, the chronic toxicity and maybe some subchronic, subacute toxicity.
Great, thanks for that perspective, Thor. Our next question is, for ISO, a score of 2 or less in cytotest is regarded as passing. However, does the FDA also have the same view, or do they prefer to have a score of 0 to be regarded as non-cytotoxic?
We've never seen a prefer of 0. We have had some reviewers question a 2 because obviously it's close to that failing line. Just so you're aware, the ISO at this point does have an acceptance criteria, but that has been in discussion to remove it because it is the only ISO with an acceptance criteria. The rest rely on scientific judgment. The concept is that you should be able to evaluate the results. A good example is latex. Latex will fail cytotoxicity. It'll give you a 4. Does that mean that that latex glove can't be used? No, you have to really evaluate that compared to historical data and a scientific point of view. So we want to kind of evaluate the failure or the scores from that point of view, and that's really what the FDA we've seen is kind of go to. But that being said, the acceptance criteria is 2 or less. We've never seen a 0 or 1 question. We have seen a 2 questions sometimes, but other times not.
Okay, great. As I mentioned, we still have many questions to get to in a short amount of time, so we'll keep going. The next question says, it makes sense to me to do the extractables, leachables risk assessment for long-term implants or devices with colorants. But if you have a short-term contact device, such as a guide wire with contact for less than 24 hours, for example, with no colorants, would there be a benefit to doing the leachables, extractables, or risk assessment?
Yeah, actually there would be, because just because it's a short-term contact doesn't mean that the colorant is not going to leach off and go into the circulatory system, depending on the guide wire, or other parts of the body. That's why it really depends, one, on your material. If it's just a stainless steel guide wire, then your extractable leachables may not be as necessary. But if you have that colorant in there, the FDA does have a high degree of concern with color additives. And so it might be, you might not, like I mentioned before, you might not need to do the whole leachable test. You might just have to say, this is the amount of colorant in the device that contacts the patient. If all of it came off at once, this is the tox evaluation. It could be much quicker and cheaper that way, and because of the limited contact, you might have a scientific rationale to do that. But if it has a colorant in it, and if it has patient contact or fluid contact that then contacts the patient, I would at least be prepared to evaluate biocompatibility or leachable extractables.
Okay, fantastic. Our next question is, could predicate devices be used to provide information on the safety of another medical device similar because of the same color additives content, or should biocompatibility testing be done in any case?
Great question. And once again, this kind of goes back to the point that the FDA gave on justification. They gave you actually kind of each thing they're concerned about when you're looking at comparing a predicate. So you're just not concerned about materials or colorants. What you're also concerned about is its shape, its function, its processing, its sterilization. All that can have an impact on leachability. So that's what you have to evaluate. You can use a predicate device to help Rationale. It just really has to meet all those criteria, or at least have a good scientific rationale on each one of those criteria to make an acceptance. My suggestion would be to sit down with some individuals, if you're looking at doing that, and really come up with a realistic evaluation and see if it's possible or not. If you can't get away from doing all biocompatibility, you might be able to get away from doing some of the more invasive animal testing.
Okay, great. Our next question is, FDA does not seem to like using a safe clinical history of a device. Do you think this will change?
Yeah, and that's kind of what we talked about in the justification ideas. It really depends on the reason they don't like the safe history of a device is they want to there's more questions there that you're not giving them. So when you're just giving them a history of a use of a device, well, has there ever been since the changes over time, or how it's used, or what materials changes? There are a lot of things that you need to question. So when you're looking at safe history of use, first off, you have to see why you're referencing that. Are you changing something? Are you adding a material that has safe history of use? And if you are, then you have to evaluate all those different endpoints that the FDA brings up. In our experience, if you meet all those concerns of the FDA, then you're able to use history of use. But just to say that my device has been used for 20 years and never had a problem, and that's why I'm bringing this material into my new device, that might not work because the FDA is going to want to see more background than that.
Okay, thanks. Does the FDA prefer the qualitative or the quantitative test for cytotoxicity?
This is a great question. And so just to give a little bit of background, I know there's tons of questions, but the cytotoxicity test in ISO 10993-5 has always historically been the MEM elution test. In 2009, we changed the standard to include some quantitative test methods, the MTT, XCT, specifically NutriRed assay. These were meant to try to eliminate the bias of the reviewer or the technician scoring the MEM. The problem we saw, though, is a lot of devices were starting to fail cytotoxicity per the quantitative method, the MTT or XCT. It was more sensitive. The cytotoxicity is already the most sensitive test. So we started seeing a lot of people go back to the MEM. In fact, we had some reviewers, when they saw a failed MTT, they requested the MEM for a confirmation test. So now their discussion in Italy was to reevaluate the quantitative, when to use it, when not to use it, what does it mean. So at this point, this is just from Nelson Labs' experience with the FDA, we always recommend the MEM elution. We have confidence in it. We know how it's run. It's already sensitive compared to all the other tests. And we have never seen a reviewer question it, at least in the last couple of years. So that is where we stand with our experience.
Okay, great. Thanks. Our next question is, have you had experience where a device has received approval using the extractables, leachables, and risk assessment to not perform some checkbox tests, such as the standard tests like irritation, for example?
Yeah, we have had lots of experience where we use the chemical characterization data to justify out of, for example, chronic toxicity, mouse marker nucleus, even where you can justify out of successfully, even subchronic, subacute, depending on some of the other information we had. And then we specifically use it for justifying out of when changes of a device. So, you're changing some of the material. It helps alleviate the need to do biocompatibility if your material, your chemical characterization comes back similar to the test device. So there are ways that we have been successful in eliminating specifically the animal tests. I want to be specific there. The in vitro test should probably always be ran, but the animal tests, we have been successful. But there are other endpoints there than just the chemical characterization. That does depend on the material, the how it's used, and other aspects.
Okay, thank you. Moving on to our next question. This is a good one that I'm sure many people are wondering. When does the FDA expect to finalize the draft?
Yeah, man, that's a great question. I'm wondering that myself too. I know that the ISO, we had a lot of response that we submitted to the FDA. Us, Nelson Labs, had independent responses, and I'm sure there's been a ton of other responses. So at this point, and I can check on that. I can contact Jennifer or some other people in the FDA just kind of get an update on what we're looking for. But I'm sure they're kind of going through that right now and trying to evaluate that and going through. But at this point, I do not have any update on that. Okay,
fair enough. We do have time for just one or two more quick questions. Just so everyone knows, I know that there are a lot of questions that we're just not able to get to. However, Thor or someone from Nelson Labs will follow up with the people who submitted the questions via email after the webinar to get some responses there. So one of our last questions, the new guidance appears to prefer direct contact for cytotoxicity. Do you agree?
That is one that I did not put up here because it was so test specific. We actually did respond to that, saying that the direct contact is less sensitive. For example, an agar overlay MEM test or agar overlay cytotoxicity test, in our experience, is much less sensitive than the extraction version. So we are not sure if it's coming across right what is direct. So we are asking for clarification on that. At this point, we're not seeing that enforced. Like I said, I did not put it in the presentation because I thought it was very test specific, but I do not agree with that. I do think that that would be changed based on the historical experience with an extraction versus like an agar overlay or direct contact cytotoxicity test.
Okay, great. And let's see, why does ISO state that chemical characterization should precede biocompatibility testing and FDA does not?
So then the ISO, the whole ISO 10993-1, the thought is that you do your chemical characterization to give you an idea of what's coming off your device. what compounds, what amounts, and that triggers what testing you should perform to help evaluate for safety. For example, if you see a chemical that's known to be a sensitizer, well, then it might trigger you to do a sensitization test. Or if you see something that is concerning from a systemic toxicity, then we might have to do some of that. But the whole concept in -1 is that the chemical characterization leads you to your test plan. The reason why the FDA, and part of me agrees with them on this concept, has kind of a different view from that, is because the amount of data we have out there for a bunch of these compounds are not necessarily sufficient all the time to make those assessments. For example, with sensitization, just because you have a chemical you identified and you don't think it's a sensitizer, there's not a lot of good data in the industry on sensitization of these chemicals. Other endpoints that might just not be as confident that the FDA. You got to remember that the FDA's responsibility is for the safety of us, and that's a big responsibility. So they do feel a lot of pressure to make sure that they have the information they need to approve a device. And for them, they just don't feel as confident, in my opinion, talking to some of them, on making that assessment yet. But we see steps going that way. Each ISO meeting, we see more and more discussion on looking at in vitro alternatives, chemical characterization, ways to eliminate the need for animal tests on devices that have been tested for 1,000 times from different people and have shown very little impact in the use in the industry. So it's coming, but that's why the kind of the differences between the international and FDA.
Thanks, Thor. There's obviously a lot of interest in this topic. Unfortunately, though, we're going to have to come to a close on our webcast for today. Again, Thor or someone from Nelson Labs will reach out to those who submitted questions that we weren't able to get to during the live webcast today. I would like to thank our speaker again, Thor Rollins, for a great talk that obviously resonates with a lot of you. We also request that you take the brief survey that's appeared on your screen. This presentation will be available shortly in an on-demand format, and as a registered user, you'll receive an email with detailed information on how you can access the on-demand replay of this webinar. The on-demand version will also include a PDF copy of the slides used in today's discussion, as I know many of you are eager to see them yourselves. Plus, more information on this topic can be found at qmed.com, mddionline.com, and nelsonlabs.com. This webinar is copyright 2013 by MD&DI magazine. The presentation materials are owned by or copyrighted by the individual speakers who are solely responsible for their content and opinions. On behalf of UBM Canon, our guest speaker Thor Rollins, and our sponsor Nelson Labs, I'd like to thank you for attending, and I hope you have a great rest of the day.